Dr. Langer's influential work has garnered him over 220 major awards. Among these are both the United States National Medal of Science and the United States National Medal of Technology and Innovation. His contributions to the field have been acknowledged through a vast array of other awards, including the Gardner Foundational Award, the Charles Stark Draper Prize, often likened to the Nobel Prize for Engineers, the Millennium Prize, the Priestley Medal, the Wolf Prize in Chemistry, and the Kyoto Prize. In 2015, Dr. Langer was the recipient of the prestigious Queen Elizabeth Prize for Engineering. Dr. Langer's research is at the cutting edge of biotechnology and materials science, focusing on the development of polymers for the controlled delivery of drugs, as well as the engineering of new tissues. His academic achievements are matched by his role as a member of multiple national science academies and his receipt of over 30 honorary doctorates from top-tier institutions worldwide. Very accomplished engineer with over 400,000 citations. His influence extends beyond academia into the realms of biotechnology and global health. Forbes and BioWorld have highlighted him as one of the most pivotal figures in biotechnology, and Time Magazine and CNN have recognized him as one of the most significant people in American science and medicine. It is definitely a treat to have Bob with us today, and it's good to have others joining in this informal mentorship meeting. Ugo, it's good to see you. So, drug delivery. We're just going to go through a few basic ideas in this presentation, and we will proceed to discuss some that's associated with some of the work we've done. Drug delivery systems, according to the National Institute of Biomedical Engineering and Bioengineering, describe technologies that carry drugs into or throughout the body. So if we do like a quick overview, we have gone from ancient remedies and herbal medicines through the pharmaceutical revolution to oral dosage forms, to transdermal patches and biotechnology and gene therapy, to targeted drug delivery and smart drug delivery systems. So you have targeted drug delivery, biocompatibility and safety, controlled release systems, nanotechnology and nanomedicine, and personalized medicine, which is a really hot topic now. So some of Dr. Langer's achievements: Bob has achieved a number of things. Some of his work has been centered around investigating the mechanisms of release from polymeric delivery systems with concomitant microstructural analysis and mathematical modeling, studying and applications of these systems, including the development of effective long-term delivery systems for insulin, anti-cancer drugs, growth factors, gene therapy agents, and vaccines, developing controlled release systems that can be magnetically, ultrasonically, or enzymatically triggered to increase rates, and angiogenesis inhibition. Done a lot of work. So some of the big themes that kind of encapsulate the work that he's done: polymeric delivery systems, controlled release systems, angiogenesis inhibition, biodegradable polymeric delivery systems, tissue engineering and biomaterials, and some key questions. So this is going to be a dialogue. So some key questions that we have for you, Bob, are: how did the work that you did help with the research you're doing now in drug delivery? Once again, it's good to have everyone on.
Yeah. Well, so really, the way I got started in drug delivery was by doing cancer research, trying to have a delivery system that I could use to deliver large molecules, which hadn't been done before, from tiny particles. So that, and of course, at the time I did this was first published in Nature in 1976. I don't think there were very many large molecules that were of interest. Today, of course, there are many, and drug delivery has been key to that. I mean, not only for delivering, proteins, but, the recent work that, one of the companies, as some of you know, that I started was Moderna, but the idea that you could deliver messenger RNA if you didn't have the mRNA, which is a large molecule, and nanoparticles, we wouldn't have the COVID vaccine, and I believe many other treatments that will come in the future. And of course, that is some of the things that we're doing today in drug delivery. We're working on delivering complex molecules by many routes, oral, through swallowable, through nanoparticles, through other routes as well.
Yeah, so we see that it advanced the field in certain areas. However, what were the lasting impacts made during the 70s, 80s, and 90s in your experience and opinion? During your early years of this work, what would you say were the lasting impacts that you saw then and that you are seeing now?
Well, I think the first impact was that you could do it at all. A lot of people didn't think you could deliver large molecules. In fact, the work we did when we wrote grants was rejected many times, and a lot of people who were experts just said that this was impossible, like trying to get a large molecule out of a tiny particle. They said either it would come out instantaneously or it wouldn't do it at all. And so, I think that was really the first thing. But then work by us and also by others, would lead to many things that I do think have had an impact. First, it's basic science. Trying to understand the mechanisms by which you can get delivery, trying to create new materials, trying to get the right kind of kinetics, different types of design. So many different things were done in the 70s, 80s, and 90s. And of course, I was fortunate. I had a lot of very great students and postdoctoral fellows who are now like professors or involved in companies all over the world. And, they did a lot of that work. And then there were lots of other people that I knew that was, got involved in this as well.
Okay, sounds good. So just before we proceed, I'll open the floor for any questions. Any questions that people have, I'll open the floor for questions, and then we'll proceed with the presentation.
Dr. Langer, I do have a question.
And you should just call me Bob. All of you should call me Bob.
All right. Wait, no, you know that I'm from the old school.
Well, you're an important person. You sir, anyway, all of you are.
Well, listen. What are your thoughts on now? You talked about nanoparticles, and typically the nanoparticles in the past were created using lyosomes, right?
Liposomes, yes.
Liposomes. Well, what are your thoughts on niosomes now?
Well, I think it's a good question. I mean, so people are making nanoparticles out of all kinds of things over the years. I mean, whether it's lipids or polymers or really anything. I think all of them are interesting scientifically. I think the issues that I would think about would be, what I always call do they have some unfair advantage for a particular application compared to what already exists? And, might depend on the particular liposome or niosome or polymerisome or whatever it is that you have to do. So I don't know that there's a simple answer, and people may come up with ways of making them better and better. Interestingly, when I started doing this, a lot of people also felt that lipid nanoparticles or liposomes were a nice academic thing, but they would never really be useful clinically. And clearly that's changed because people figured out better and better ways to make them and better components. So, and I think that's one of the great things about science. You can keep evolving it and making it better and better. So I always hesitate to say what's better. I think all these things are good, and can have potential advantages.
Okay, so go ahead, Dr. Tin. Do you have anything else you want to ask?
No, we'll let that go.
Okay, yeah, that's good. Okay, so moving right along. The presentation was composed based off of the extended list of articles that I found for that Dr. Langer played a part in writing or was an author in. So they were organized by years and segmented by each 100 in terms of how it was arranged in the list, and the themes highlighted that covered the ideas. So if you look at the early portion of Dr. Langer's of Bob's work, we see that many principles, technologies, and concepts he developed during the 1970s and 1980s continue to be relevant and are actively applied in the design of modern drug delivery systems with a focus on improving therapeutic efficacy and patient outcomes. So some themes. So these are some from Bob's early work. So you had enzymatic regeneration of ATP in 1974 and 1976. Then you went on to the pH-dependent assays, mast cells and tumor angiogenesis, polymers for sustained release in 1976, the isolation of a cartilage fracture in 1976, and continuous long-term intra-arterial infusion in 1978. Moving right along, his work that continued into the controlled release of macromolecules for polymers in 1980, zero water release kinetics in 1980, implantable drug delivery systems in 1981, microspares for controlled drug delivery in 1984, and local controlled release of diphosphonate in 1985. So, the question posed to Bob and to you is, would you say that your work with Judah Folkman really laid the foundation for you to do this work that you did in the early portion of your career? Or would you say it was your graduate work and the work, your postdoctoral work with Judah Folkman that complement to the progress, advancement of drug delivery research milestones during this time?
Yeah, I would say that it was definitely the work with Judah Folkman. I mean, he, partly because of trying to solve the angiogenesis problem, and partly, because, and then the work with him, and then the later subsequent work at MIT that I did with different graduate students. But that was really the key things.
Okay, sounds good. Okay, moving right along. So the questions that we have: what were the foundational principles that guided your research inquiry during the 1970s and 1980s? What were like the overarching questions that you were trying to answer in the 1970s and 80s? And are they still influencing your current practice in the field?
Yeah, so I would say that, part of it, it was trying to understand, since people told us we couldn't deliver large molecules and I felt we were doing it, there were a couple of things. One was doing it and doing it very reproducibly. A second was truly trying to understand the mechanisms. A third, we're developing newer and better materials. And I'd say some of those I feel we've solved, but they still influence what I do today. I mean, we continue to try to figure out, how to make better systems and solve new generations of problems. I mean, now that we have the foundation to make nanoparticles or microparticles or just deliver large molecules in general, you always want to do better, right? Can you target them to specific cells? Can you cross barriers in the body that are tough to cross, like the intestine or the brain? But I think the foundational principles were.
But any thoughts on that, Aaron?
Yeah, absolutely. Well, one of the ones. I mean, there are other drugs like Alkermes, which was based on some of our work. They've been using, I think it's now naloxone or naltrexone. But you're right about buprenorphine. I mean that right now is like in a sublingual system, and I believe some people are developing on injectable ones, like, and maybe that's what you meant, where they're sub-Q or intramuscular. I mean, I'm not an expert. I don't know what all the issues are. I mean, I think some of the things that I know people have talked about are, could you make them last longer? I think that's certainly possible. I know I talked once with Richard Pops, who was the CEO of Alkermes, which is one of the producers of the naltrexone one, and he said, that's a 1 month system. They'd like to make maybe someday a 3 month system or a 6 month system. So I think developing them longer probably would be, one of the things that would be useful. That particular system is called Vivitrol. But, and I would think that those kinds of principles could be extended to buprenorphine, but I'm not expert enough to know all the issues with buprenorphine.
Vivitrol has worked well. I still think that there are some challenges with therapeutic concentration and maintaining that therapeutic concentration.
Making it flatter, you mean?
Yeah.
Yeah. Well, that's a good question, and that I think is something that could be done, by I actually mentioned some ways long ago. I was actually on their board maybe 20 years ago, and I agree with, I think the ways to do it, you could, if you make sort of the effective particle size very, very tiny, I think you could probably help on that. And the other thing, though, maybe it's not a good idea from a manufacturing standpoint,